the Dementia Researcher podcast, talking careers, research, conference highlights, and so much more.
Today we're in Liverpool to bring you highlights from the UK's largest dementia research conference run by Alzheimer's Research UK. I'm Dr. Yvonne Couch. I'm an associate professor at the University of Oxford, and I work on stroke and extracellular vesicles and vascular dementia. And today I am joined by three amazing guests to talk about the highlights from the ARUK annual conference. We've got old hand Jodi Watt, who writes regular blogs for us, and we've got two newcomers, Becky Carlyle and Yasmin Bonsu, who are going to share with us what they really enjoyed from the ARUK conference. Say hello, guys.
Hi.
Hello. I think it will be useful to have an introduction from everyone. So Jodi, you're a regular blogger, so why don't you go first?
Sure. So I'm Dr. Jodi Watt. I'm a postdoc at the University of Glasgow, where I work with Terry Quinn on drug repurposing currently. And I'm also coming at this from a clinical perspective.
Excellent. So Becky, we chat all the time, so I know what you do, but why don't you tell our listeners what you do?
Yeah, so I'm Dr. Becky Carlyle. I'm an Alzheimer's Research UK Senior Research Fellow, and my work is focused on cognitive resilience. So some people get a lot of amyloid pathology in their brains and they have very severe cognitive symptoms. Others barely experience any cognitive symptoms at all. So I use techniques like proteomics to try and find out what's different about the brains of resilient people versus susceptible people.
Excellent. And Yasmin, I know you've been doing some work with Becky. Can you tell us a little bit about your background and what you've been up to in the lab?
Right, well, I'm Yasmin Bonsu, and I'm an undergraduate student from the University of Manchester, and I'm currently doing a placement year under the supervision of Dr. Becky Carlyle. I am looking at neuropeptides from the granin family and how they could possibly promote resilience in cortical neurons that are insulted of amyloid beta.
That's very cool. Excellent. Thank you. And thank you guys for joining us. For new listeners, let me explain the format of the podcast. So it's quite simple. Essentially, we're going to go around 3 or 4 times. We're going to talk about each day of the conference. We're going to say some things we like. We're maybe going to talk about some things we didn't like or we found a bit controversial. And we're going to start with the ECR day. Now, fortunately, all my guests today were at the ECR day. So we're going to start there. And Yasmin, you're our official ECR guest because I feel too old to be an ECR now. So I don't count, but you're definitely an ECR. Tell me what you loved about the ECR day, or what you hated, or what you found interesting.
Okay, ECR day, I liked how there was so many research assistants, postdocs, PhD students, and it was a way for me to network and basically ask people how they got to where they are. Like as an undergraduate myself, I'm questioning what do I want to do next. So it was a good way for me to get some more information about what the next steps could be. Like, should I do a master's and then go on to do a PhD? Should I just skip master's altogether and then go on to do a PhD? So it's good to get insight on that. Additionally, I was really moved by the talk from Alison and Frank, that couple. Which is really beautiful.
It was.
I wanted to cry, but I didn't want to ruin my makeup. But yeah, I was really touched by their story, and it just made me really special to be a part of an organisation that's helping them and others as well.
Yeah, and they do it every year, and it's always it sort of puts your work in context, but I was unprepared for it. So last year was my first in-person one, and I was blissfully unprepared for it. I sat down and I was like, what is this? And I was just weeping, and it was awful. And you cunningly hid in your hotel room so you could cry in public and nobody noticed.
Every ARUK conference now, the first session will be taken online at home. So that I can cry as much as I need to cry, then put my makeup on and go do the rest of the conference.
It's absolutely devastating, but I also think it's really motivating as well. You're like, I know everything that we do sometimes seems quite removed from that, but it does feel like you're, making a contribution. So I'm glad that was your favourite bit.
Yeah, it was.
I think that's something that also, as someone who works clinically, I think that's really important as well, because a lot of conferences that I've seen that have a bit more of a sort of preclinical tone to them often sort of miss out having the sort of patient representation or familial representation there. So I was on the early careers day, I was really struck by Jordan's talk. so Jordan is somebody who has familial history of fronto-temporal dementia and also carries a gene for it. And I think the things that he was doing are just incredible. And it was really nice to have that perspective as well, because it's something at the talk he mentioned before was, I was nearly crying as well. But I was also nearly crying at Jordan's, because I think we often focus more on the old age aspect of it. And he's younger than I am, and he's facing like that sort of thing and that sort of knowledge about himself going forward. And I just think it was incredibly and very, very inspiring talk. Yeah.
I think it's really nice to have that. We've got our local network coordinator, Katie, she's here, and she fundraises tons for AI UK. And it was so nice to have the fundraisers here as well, and so you can show them what you do. So I took Katie over to my poster. It was so much fun to go, you ran, however, I think she ran a marathon or a ton of marathons.
3 marathons.
3 marathons.
Yep.
She ran 3 marathons. And she raised way more than 5K, but I got 5K and I was like, this is what it's done. And it's launched another project, and that's got me a job for 2 years, and it's amazing. Yeah. So I think that sort of feedback between the people who have the disease and the people who are giving the money and the people who are doing the science is really, like, I think it's done really well at AI UK.
I think there's definitely a tone of like, from those sort of speakers about how it felt like it gives them purpose. And I think you root that purpose in something tangible, and I think that's really important as well. And I think AR UK does that really, really well.
Exactly. So Becky, you gave a talk on ECR Day. Did you get good feedback? Did you enjoy it? How did you find it?
A lot of people enjoyed it. Not too much specific feedback, which is good because it just came out last week. But yeah, I think I managed to get across that my lab is a fun place to work, and I hope a supportive environment. And I think that is just as important as the science that we do as well.
Yeah, and I think, like you say, getting your name out there and going, this is what I do, is so useful for like networking and stuff like that, because suddenly everyone goes, oh, she's the proteomics lady, I'll go talk to her about that. And you get known as a thing, and people point other people to you, and it's a useful way to put your name out there, especially to younger people who might want to come and work for you.
Yeah.
Cool. So that was the ECR Day, but the ECR Day format is not necessarily the same as the rest of the conference. It's definitely much more focused towards younger people and showcasing their work. But the main conference is obviously much more sort of a traditional conference with sessions with different focuses. So we'll talk about that next. So day 1, Jodi, we'll start with you. What did you enjoy about day 1 of the conference?
A good question. I surprisingly, to my general being as a scientist, I actually really enjoyed the AI debate. I'm to set tone. I am very skeptical of AI's involvement in things like diagnostics, because I know the sort of black box element of things for people who don't intrinsically understand AI or have to do a bit of, I just trust that this thing works, is really scary to patient populations. And obviously working more clinically, I sort of am more aware of that sometimes than people who work preclinically. What was a real highlight of it for me was the fact that although everyone involved in it was clearly in the world of AI to have the knowledge base to do it, it really highlighted for me the scope of their knowledge and the understanding of their knowledge in something like AI, which when you're someone who's trepidatious about it is really reassuring. And as someone who has sort of certain minority characteristics, which I've written about at length, so I won't bore you with just now, it was really reassuring for me to see that they recognise those. And it ended up being a really interesting space where almost to win the debate, they were having to discuss that at length, and that doesn't often get, that's often the end sentence of we know we need to do this better on a poster or an abstract, and it's really good that got airtime even though that wasn't the direct topic. And as a skeptic who voted and was in a very low proportion of people who voted against the motion for AI at the start, I really enjoyed that was the motion that ended up winning out.
Yeah, because I think it was only about 10% at the start, wasn't it, who said they thought it was a bad thing, and then it went up to like 60%.
Yeah. So I felt vindicated.
Not
that it was about me, but yeah.
No, I think it was really nice, and David Llewellyn was so... he was very eloquent about, I liked his phrase about bias getting accidentally baked in.
Yes.
And the risk of that happening if we just start using it now without thinking about what we're programming into it and stuff like that. I liked that because it was a bit of a debate, There was a lot of AI in this conference, though.
Yes, that's fair comment.
I don't do AI. I don't do big data, so I found it baffling and ever slightly boring.
We didn't actually even talk that much about results that AI has generated. We talked about building platforms for it. We talked about a little bit about the basics, but not really enough for anybody to walk away thinking, oh, I understand this now.
Yeah, exactly.
And, we didn't really talk about use cases. And, we could have had a talk on somebody using it to do some kind of image analysis or, a new diagnostic pipeline. We didn't see any of that. We just had these kind of scare stories and debates, but nothing about use case.
But this harks to the black box idea, right? That patients are, and people living with dementia, I should say, sorry, like their fears around that sort of thing. So yeah, I'm glad that was picked up on, but I don't feel like I left the debate knowing what we do about that.
No, but I think that's one of those big life decisions that everybody is slightly terrified to make. And
Sure, absolutely. I don't have an answer.
No, I don't have an answer either. I think somebody needs to make that decision at some point, but I don't know how you go about doing it. It's kind of like the euthanasia thing and just worse. But yeah, I had a discussion about the multi-omics, the session that you were in. And there are a few people who were like, it would have been really nice to make that more of a, how can we use all of these techniques to answer more questions rather than, this is my technique. Not that it wasn't bad, your talk was lovely, but I think there was scope to make it a bit more, not inclusive, I'm not even entirely sure what the word is, but like a bit more sort of, let's think about how everybody could use these kind of things.
Yeah.
But it was interesting, and I did enjoy the debate. I liked watching it, and I definitely switched my mind because I was like, yeah, AI sounds great. I know nothing. Becky, tell me about your highlights from the first day.
Well, that actually leads on. So I think my highlight is a very strange one because it was a very dry talk, and I hope Vanessa forgives me for saying that. But I think Vanessa Raymont's talk was very important because that shows exactly that people are working really hard to build these platforms that you're talking about. So, her role as Dementia Platforms UK lead and in building together the network of brain health centres from across the UK, like those are the kind of setups that we need to be able to do these kind of studies ethically with the power that they deserve and to have the ability to integrate these data sets across different modalities. So forgive me, Vanessa, it was a bit of a dull talk, but what she's building and what she's collaborating with to do is really critically important, and to get slightly political, should be government funded now. Like they're struggling to scrape together pots of money from all over the place. This should be a central thing that the government is trying to fund, because with the structure of the NHS, with the work that they're doing, we could be streets ahead of every country in the world on this. We have a more diverse population than most of the developed European nations. We can maximize this, and we can really be the leaders in this space. So I can't say it was the talk I enjoyed the most, but I think it was the most important message for where we're headed. And if we get the money if they get the money that they deserve, then the future that we should have in dementia research in this country.
Yeah. Oh, it's all about money. It's somebody said at some point about funding bigger picture stuff, and he was like, it's not all about the money. I was like, yes, it is. It's all about the money. You can't fund this kind of research because it's all longevity. It's all like, nobody gets dementia and then dies the next year. I mean, some people do, but it's much rarer. It's a very long progressive disease, and so you need people who can monitor over that length of time, and you can't do that with a 4-year fellowship. It's just not possible. So more money would be better. Government, more money, please. Yasmin, tell us what your highlight was from the first day.
The first day, it would have to be the talk presented by Dr. Soraya Mehta.
Yes.
That process, like her methodology of what she was doing was insane. She was using live human brain slices and then cutting them up and putting them into culture medium. And then they were characterizing like Alzheimer's pathology and looking at how endogenous amyloid beta is affected using an inhibitor called BACE1. I can't remember the actual name of it, but BACE1. I thought just using an actual human brain was pretty cool. Like you've got post-mortem samples that you could use, and that's in itself isn't pretty amazing. But to use someone's actual real brain and then carry on to like create a more translatable model to study a neurodegenerative disease, it was pretty impressive.
Yeah.
I mean, I can imagine it's an expensive process, but a cool process nonetheless. And I was very inspired by it.
Yeah, it was really cool. I really enjoyed, though, the questions that came after her talk. There were so many questions because it was obviously a really popular talk, but I think my favourite one was, do you think these slices can still think? It's like half of your brain is in a dish somewhere going, help, let me out. I was like, that was the best question ever. So I will briefly give my highlights from the first day and then we can move on to day 2. I really was anybody in the session in Auditorium 2 where Ramona Weil talked? So this is far outside my comfort zone, but she gave such a good talk. So she does visual stuff, and it's all about the progression of people who have Parkinson's but progress to get dementia and go into cognitive decline. And that doesn't happen in all Parkinson's patients, apparently. And one of the things she's found is that the people who do progress to dementia have early visual changes that you can see. And so she's gone and done this huge battery of visual tests. And my favorite one, they called the cats and dogs test, which is they take a picture of a cat or a dog and they basically slant it to the point where it becomes almost, she put one up and I'm there going, I don't, she didn't say it was a cat or a dog when she put it up, and I'm looking up at it going, I don't even, what is that bunch of grey blobs? And then she said, it's pictures of cats and dogs. I was like, oh, it's a dog. I've not got dementia, it's fine. So yeah, and apparently the people who are more likely to progress to dementia find those slanted pictures really, really difficult. And so she's now got all this sort of battery of visual tests to use as almost like an early biomarker, which I just thought was very cool. I was like, it's very easy to do. You're not sticking needles in people. You're not putting them in expensive magnets. You're just getting them to look at pictures. And you're able to say very early, this person is more likely to go down this path. So maybe we can jump in. So I just thought, and she was just a very good speaker, which I always appreciate at conferences. But then we had day 2. So, Yasmin, we'll start with you this time. Tell me what you enjoyed about today, essentially.
It would have to be the talk we had about in healthcare. There's like a lot of disparities when it comes to diagnosing people with dementia. There's currently some resources that are available right now. I think one of the ladies that was presenting is called Mila Kavipeto. She has founded a program called the WW Fingers, and essentially it's looking at trying to diagnose people earlier on across all demographics and across like all geographic locations. I thought that was pretty important because today I would agree that when it comes to ethnic minorities, they feel very reluctant in going into the GP or to the doctors to address their healthcare concerns. They feel like they'll get dismissed or they won't get their problems gets viewed at properly. So it's nice to know that there's people in science that are doing their best to kind of address that, inequity.
Yeah, it's still a massive problem with women going to the doctors who are like, well, I've got this pain. And they're like, oh, well, you're a woman. It's probably just period pains, when it's clearly not. And there's a lot of stigma. And there's a lot of stigma about going into, it's the same with mental health. There's a lot of stigma about going in and going, I'm getting a bit forgetful. Like, you don't want to feel like you should go and see someone, but actually you probably should.
Yeah, one of the panelists, they mentioned a stigma. I think it was in Nigeria. Like, some patients don't want to actually be classified as I have dementia because then they'll get branded as like a witch or something like that. And I didn't know that. So that was quite surprising to me. Like, there are stigma around having an actual disorder.
Yeah. And especially when it's brain stuff, I find that people don't, a tumour is a big thing that grows on you and you can kind of see it or you can see it in a scan. But your brain is still, I think, a bit of a black box. And so people, when stuff goes wrong with it, people get very uncomfortable.
That's so true.
Not good. Jodi, tell me what you enjoyed about today.
So I'm going to take something that was slightly off conference plan because it was at lunchtime.
Ooh.
And I was expecting it to be low attendance, and I was actually incredibly surprised at how many people kept coming in. So it was the Women in Neuroscience talk, which is interesting for me as someone who falls down a gap of gender and doesn't really conform to that. But I wanted to go and I wanted to see their plans, what they're doing, how they might be able to fit someone like me into their narrative, because obviously when you don't identify with that, the word woman is quite intense as a choice. But I had spoken to, I can't remember her surname, but Lizzie
English. Possibly. She's the main WIN person, I think.
Who founded the whole thing. And I was really impressed with her, so I went along, and I just feel very excited about what they're planning on doing. I also really enjoyed, yes, I didn't see myself on the panel, but I appreciate that having someone who's non-binary or gender non-conforming is tricky without being tokenistic. But there was a lot of diversity in the women that were on the panel. And there was also a lot of transparency, which I really appreciated. It was very nice to hear people, 2 of whom were professors, talk so openly, but very carefully isn't the word, but they were very pointed in what they were saying, but they were being very open. And I think that was really nice to hear people so senior be like, yeah, it's quite bad. It's not great about gender. It's not a great thing. So I was actually really impressed with that, and there were so many questions, and I was a bit like, oh, my question didn't get answered. But at the end, Lizzie said that she'd taken note of everyone's questions and she's going to address them all. So I actually felt very, very heard in that space. So although some of it isn't great for me, I feel like there's active effort to change it. And I think that thematically has been something for me that I've noticed in the conference, that where there are issues like that, people are trying to change things, which is exciting. And I just like massive shout out to her, because she's really doing a great job, I think. So.
Definitely. I think we've had this conversation a lot at Oxford about, it's almost like you don't I'm not saying you don't need more women in neuroscience, that's definitely not a thing, but we've now got lots of people at our stage, but it's getting lots of people above us, and it's nice they have female professors, because you can then see that it is a thing, but it's still not enough of a thing. And I think I'm hoping that's what's going to be the next stage of all of this.
Yeah, it would be nice to see some sort of development with regards to other genders and their representation, because I was having this conversation with someone earlier, like, people have referred to me as a role model, and that's fine, but I'm an ECR. Like, I'll happily be it, but I am an ECR, and normally when we look at academia, we're talking about professors who've done like multiple Nature papers or something like that, or had a huge discovery. And as someone who's ECR, where's that person for me? You know, that's tricky. So what I took from that was actually, she was so good, she might be the person to do it. She might be the person to get the representation that I'm looking for, which is incredibly impressive, because I'm used to going into conferences and being like, nope. But actually,
none of this is working
for
me.
once, I was like, maybe. So,
can I ask you a personal question? Do you have a role model, or are you working as your own role model?
It's an interesting question. I think that what I think a lot of people look for in science is a direct role model, right? And I definitely don't have that. I would say that there are a bunch of people that I work around who do things in such a positive way that they are inadvertently my role models. But in terms of like how I hear people talk more traditionally about role models, like people being like, this female professor is incredible, I want to do what she's doing, I want to have the success that she's doing, and she's managed to do it whilst also having children, which is an annoying caveat to things, but one that is touted a lot. That sort of thing I don't have, and I almost have to be it for myself, otherwise what am I going to do, you know? Exactly. Because I have to find it somewhere, and it's been pointed out to me that it's one of my strengths, and that's advantageous, but it is tricky to be like, oh, I have to be this for myself. Okay, cool.
Let's just roll with this and see how it works.
Yeah.
And it is meaningful, isn't it? You know, it's easy for somebody who's a white male to just bat it away. This isn't important.
Yeah.
But Yasmin, you met Yasmin Hurd a couple of weeks ago, right? And that was, I mean, talk about the experience for you.
Oh, she was absolutely delight. Like, she was talking about her work in addiction, and she's a professor at Mount Sinai. And after her talk, I was a bit scared to go up to talk to her because, I mean, like, she's a big name and I'm just regular Yasmin. But when I spoke to her and expressed how scared I was to, like, come up to her, she was like, "You don't need to be scared. I'm just like you." And she gave me helpful advice in, you know, how to progress in my career. And the number one thing she said was to network. And that's what I carried with me today. And this week, actually, So it was nice to talk to her.
Because presumably, and this is, again, you might be a little young maybe to have a role model, but but there are not a great deal of sort of people of colour in senior positions.
Exactly.
Do you think that's a problem, or is there someone who you see as that kind of role model for you?
To be honest, before I met Professor Yasmin Hurd, I didn't really have a role model like that. I wouldn't say it was a problem because I think in the academic field, I wasn't really surprised that there's not a lot of people that look like me in higher positions. So I wasn't going to have like my hopes set to have, oh my gosh, I need to see someone like me that's a professor or, you know, a senior doctoral. But to see Professor Yasmin Hurd up on that stage in Oxford giving a nice lecture to lots of people, I realised I can look for other role models now. So I am going to start doing that. But as of right now, she's my number one.
Excellent. Obviously. Obviously, Becky is everybody's role model, including mine. Becky, if I asked you your highlights? Now we've got... Tell me about your highlights.
And I have some science highlights.
Excellent. Tell me your
today.
science highlights.
So I thought the science was much, much more interesting today. Like, we had real experiments, real data. In fact, I have too much competition. So I thought Chloe Anderson, the person that won the ECR Talk Award through the drug repurposing, thought that was really, really great. I would really love to see her extend it to electronic health records and see if any of the drugs she found have got any data there. Ryan Bevan was brilliant talking about PLC gamma in the inflammation. But I mean, Saba Siddiqui, it was almost like somebody had told AI to write the talk that would most appeal to Dr. Becky Carlyle because it hit all my favorite techniques. It did it so elegantly. I think she did a really good job of talking about how they fit together, how they built this puzzle about the cryptic exons and whether they produce peptides in TDP43 pathologies. And then she did a really, really great job when people at the end were kind of asking her, like, what are they for? What are they about? Of just talking about the uncertainty. Like, yes, this is a possibility, this is, but accepting that we don't know what they're for yet. We don't know if they do anything, but they definitely exist. So, you know, we've got a biomarker maybe, but who knows what else might come from those. And it was such a confident presentation. It was so well put together. And I mean, ribo-seq, proteomics. I mean, it really was as if somebody had written my dream talk.
You're such a nerd.
And then done, I know. And then she did an incredible job of presenting it. So,
she did. It was really good. And she was about the fourth person who'd mentioned cryptic exons. And she was the only person who actually explained everybody just kept dropping it in. And I was going, I don't know what a cryptic exon is. And she told you, and I was like, she had a diagram. I'm a big fan of a nice, clear talk. And yeah, we did actually have a surprising number of those. There weren't that many people who just leapt in and went, here's all the science. I think there were quite a lot of good not, in my opinion, quite enough of them, but there were quite a lot of good talks. My highlight was mostly because I did my highlights really early in the day was Siddharthan Chandran, the director of the DRI, who spoke right at the beginning of the day. And the reason I liked his talk, and again, it wasn't particularly sciencey, but he highlighted all of these big things that we're not. At one point he said, people age at their biological age happens at different rates, and is that a good thing? Is that a bad thing? Why does it happen? And then he just went, we don't know. And I was like, great. So I really like it when people say, these are the questions and these are the answers, and this is what we don't know about a subject. And I think people are so, at conferences especially, people are so keen to go, this is my data and this is what it means, and there's not enough of, actually, this is my data and I don't know what it means, and here are the next things that I could do to find out. I think embracing uncertainty is a bit more fun.
I think that's been tonally interesting about this conference, because I definitely come from a world where people are like, here are all the positives about my science. And then when you go, like, when I say this, I mean like my PhD sort of time, when you go, yeah, but what are the problems? They go, no problems, like that sort of thing. And I definitely think this has been good at people, like this conference particularly has been good for people being like, yeah, I don't know.
Yeah.
When it's appropriate to do so, you know.
Yeah.
But I think that's really important because that sort of thing is like imperative to our understanding, where rather than bluffing and confusing, it's so much better to just go, no idea, to be honest.
And I love it. I find that at we should chat very briefly in a second about the poster session, but I find that poster sessions are really good for that because you're stood in front of someone, and whilst you've got in front of you the optimized peak version of their data, because you're chatting, they will often go, oh yeah, but we tried that and it didn't work, or we tried this, and it becomes a little bit more realistic in what we all experience in the lab. And yeah, did anyone, this is off the cuff, did anyone have a favourite poster that they enjoyed?
Other than my poster.
Other than your own poster, obviously.
Something that I didn't fully comprehend about ARUK, and it's my own fault coming in, was how preclinical this specific conference is. So my real interest, because of, you know, different fields and a slight lack of understanding, was in predominantly the clinical stuff, but I had some very interesting discussions about some of the results that I was seeing in relation to what was being seen in like cell culture in the clinic, not in the clinic, in the lab. And I thought that that was particularly interesting. I can't at the moment remember the name of the person who did that, but I really enjoyed their poster. And I love a good clinical poster, and I think there were a lot of them. There was one next to me which was focused on, I'll absolutely butcher attempting to remember the title of it, but it was focused on ethnic disparity specifically, was I think the terminology they used in dementia research. And I think something like that's so fundamental and so important. But when we talk about different types of science, or sometimes when we go up the scientific hierarchy to some of the professors and stuff, they're so focused on the cutting edge that they sometimes forget about that sort of thing. So it was really nice to see that represented at a conference like this.
I loved the amount of, I think nobody's mentioned Tamara Lashley, which I'm devastated about because I assumed somebody else would take her as a highlight. So I will mention her name now because I thought her talk was amazing. But one of the reasons I thought her talk was amazing was because she put up some nice basic immunohistochemistry. Like, here I've stained some brains, and like you say, there's so many people doing all this cutting edge stuff that sometimes I do a lot of basic immunohistochemistry, and sometimes I feel like I'm sort of almost like a chiseling in the Stone Age, and everybody's judging me for being a bit backwards. But actually, it's still being done, and I think if it's done well, and hers was just beautiful. It's really nice, like you say, to see that put out there, being done and being done well. And she's lovely, and she was my favourite bit about her talk was her very end slide, when she went a little bit crazy about all of her alumni and how proud she was of them, and like where they've all gone and stuff, and it was just great.
I think her talk, and also the poster session generally, highlighted for me as well the purpose and the utility of back translation, because I think from a clinical perspective we can sometimes be a bit like egotist about how preclinical people need to translate, they have to translate, but then when people are like, well, you need to back translate, we're like, no thanks. And that was really highlighted for me as well, when I see that's I think how I find talks about preclinical stuff engaging, when I can recognize my work in it, which is sometimes a struggle, particularly when people don't explain what things are, like you mentioned.
And particularly when it's really, really niche molecular biology, like how does this relate to a human? I don't understand. Yasmin, this would have been your first poster session. How did you find the experience?
I thought it was, what can I say? I thought that it was a good experience for me. I got to have a couple of comments. Someone recommended that we could try using a cell line that creates its own endogenous amyloid beta. So to get like feedback like that, I realized how important it is in order to develop your work. Like before in university, when it comes to like, you know, doing practicals, you don't really have a lot of attention to it. Like you're just following mindlessly the instructions of what to do. And you don't really have any like you don't feel like there's any worth to you, if that makes sense. But with your own research, it's like your baby. So you want to try and find everything you can to like make that baby grow into the best possible version it can be.
Excellent.
And that comment, like it helped in doing that.
Yeah.
And it was also a good practice for me to become more confident in speaking to other people, especially those who are like, you know, smarter, more advanced in their field.
Did you talk to a load of people at their posters?
Yes, I spoke to, not loads, I spoke to a fair few. Even got a phone number of someone who I can always just chat to if I actually need help.
That's amazing. I think it's really useful, especially at your stage, to just bump into people at that. I went up and I was an old professor at somebody's poster, and I'm not going to remember her name, which is terrible, because she was absolutely lovely and the way she presented her poster was amazing. And she was working, so if she's in the unlikely event she's listening, she was working on the 5XFAD mouse line, and she was looking at depression like stuff, but induced by sickness behavior. Induced by inflammation, and sort of inflammation and sickness behavior and depression. That was all my PhD stuff. So I got all on my high horse and I was like, what is this? Explain your stuff to me, because I think this is this. And then I went at her with opinions. But her work was great and she could explain it all really well. And she had some beautiful, basically, I remember what she did. So she basically showed that if you made these mice slightly systemically inflamed, they ended up with fewer plaques. And we think it's because you sort of stimulate the immune system to, like, you G it up a little bit and it starts munching things up early. She hadn't done it sort of super long term, but she looked at it after, I think it was maybe about a month or so. And it was really interesting data. And I feel very bad that I can't remember her name, but she was very good. And I really enjoyed chatting to. I like chatting to young people. It makes me feel young. How was your poster session, Becky?
It's always my favourite part of a conference. I just love those conversations. And I think, you mentioned earlier kind of this is your best, most pristine data. I mean, I actually, when we were talking about planning your poster and you were like, I don't have any data yet. I was like, it doesn't matter because a poster is a conversation starter, right? What you want is those people who've done these kind of experiments to come to you, and then you can have those kind of conversations. And so yeah, I think that's it. I love those conversations. It's where genuine collaborations get made. And yet there were just so many good ones. I couldn't pick a favourite.
No, they were. It was a great poster session. I really enjoyed it. So we're almost out of time, and thank you for everyone who's listened. So for those of you who couldn't attend the conference, all the details of all the sessions are on outsres.uk. We're almost out of time, but before I finish, I want to ask you guys one more question. So based on all the stuff that you've seen, what are you most excited about in terms of your research and dementia research in general? So Becky, we'll start with you.
The ability to start doing these really, really large multi-omics data sets, and the fact that people way, way above us are building those frameworks now for them to happen.
Excellent. Jodi?
For me, slightly selfishly, it's that people know about equity, diversity and inclusion and are trying. And I think that that's half the battle. So, yeah.
How about you, Yasmin?
For dementia research, I'm looking forward to a more personalised approach when it comes to helping people find a treatment. Like you mentioned, Chloe Anderson and her sex stratified approach in diagnosing, like you mentioned, looking into health inequalities. I think if we take that into consideration when it comes to diagnosing with people with dementia, that seems a bit hopeful. And regarding my own research, I would like to talk to Becky about this. I'm looking forward to going into more detail about that cell line that does produce this endogenous.
She's pitching you science ideas now.
It's impressive, isn't it?
You have no choice. It's on the podcast. It's been said out loud now. You have to do it.
Well, whoever this mystery person was, they also told Millie the same thing.
Yeah.
So, whoever you are, if you're listening, we're up for it.
I think they share the same supervisor in Manchester. So yeah.
Oh, one of the Manchester. There's tons of the Manchester crew.
Yeah.
So just to attack them, they've got loads of money, as far as I can tell. I feel like I should add one. I was quite enthusiastic about, because I'm a vascular person, and like somebody presented at the beginning of possibly today, somebody said oligodendrocytes get a bit left out. I'm not excited about oligodendrocytes. I'm sure they're very exciting, But I feel like sometimes the vasculature gets a bit left out. And I feel like there was more vascular stuff at this conference than there has been previously. And I like coming here because you knock into so many people who are doing similar things to you, and it just makes you feel a little bit inspired about going back and doing your own. Like, I need to write a grant on this, and I need to finish this paper, and I need to show these people this data. And it just, I don't know, you come away feeling a bit more enthusiastic about your science than maybe you did before, especially given that it's been January and February and it's been grey and cold and miserable. So that's all we have time for today. I'd like to thank our amazing guests. So we have Yasmin Bonsu, we have Jodi, and we have Becky Carlyle. I've been Yvonne Couch, and I've been your host for the day. And don't forget to click those like and subscribe buttons wherever you happen to be watching this. You've been listening to the Dementia Research podcast live from Alzheimer's Research UK in Liverpool.
The Dementia Researcher podcast was brought to you by University College London with generous funding from the UK National Institute for Health Research, Alzheimer's Research UK, Alzheimer's Society, Alzheimer's Association, and Race Against Dementia. Please subscribe, leave us a review, and register on our website for full access to all our great resources. dementiaresearcher.nihr.ac.uk.